Hi everybody, today I'm going to begin the second part of my lecture on membrane transport. Remember the first lecture dealt with passive transport. This one I'm going to deal with the active transport mechanisms. And I'm going to begin with the sodium-potassium pump. While there's many other pumps in our bodies that control the movement of ions in and out of our cells, the sodium-potassium pump I feel is probably one of the easier ones to understand. Very important here in understanding how certain cells work. Since they are found only in animal cells in particular, it's very important in returning the nerve cell to the resting membrane potential so that the next nerve impulse can proceed. So this is an important mechanism for controlling all nerve impulses. In animal cells, what you find happening is there's a higher concentration of sodium outside the cell and a higher concentration of potassium inside. And what that generates then is obviously it creates a concentration gradient. With the ion channels here, sodium does leak into the cell, and potassium leaks out because of the concentration gradients. But in order for a nerve cell in particular to fire an impulse, you need to have a higher concentration of sodium outside. You don't want those concentrations to reach equilibrium. So nature's evolved a sodium-potassium pump. So now what they're going to do is they're going to use a small amount of energy in the form of an ATP molecule that's going to change a carrier protein and actually pump out, 3 sodium ions, pump them from the interior to the outside, at the same time pump in 2 potassium ions. Now the next slide we're going to show you how this all works in step-by-step progress. This is a showing, 6 steps here showing how the sodium-potassium pump works. First, we have these carrier proteins. This is a carrier protein right here, and all proteins have a certain shape. And this one is specifically shaped here to fit in sodium ions. So the first step here, and since this is a cycle, we can really start at any place. 3 sodium ions bind to this carrier protein. Now, once they're bound, next step here, a small molecule of adenosine triphosphate, ATP. ATP with 3 phosphates comes along and bumps into this carrier protein, and as a result it transfers a phosphate group. So adenosine triphosphate becomes adenosine diphosphate, the low energy state. This process of bumping into this carrier protein changes the shape. Once the carrier protein changes the shape from the addition of this phosphate group, the shape changes and those 3 sodium ions are pushed to the outside. Now, once they're on the outside here, it all sets up here for the proteins now to accept the potassium ions. Now, when the potassium ions bind, it kicks off the phosphate group that was there, and that kicking off the phosphate group again changes the carrier protein shape. And when the carrier protein shape is changed, those potassium ions flow in. So now we've got a higher concentration of potassium inside the cell and a higher concentration of sodium outside the cell. And again, this is important here for such things as the achieving a resting membrane potential in a nerve cell. Showing the entire process.
Sometimes a cell needs to move a solute against its concentration gradient. This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Now remember, while we're pushing out the sodium ions into the outside here, not shown on here, there's other carrier proteins, gates of here, ion channels, throughout the plasma membrane. So these gates are going to allow the sodium to leak back in. So it's going to get pumped back out. It's kind of like a hole in a leaky boat. You know, if the boat has a hole in it, water slowly seeps in, and when water seeps in, somebody has to bail it out. So it's always going to be coming in, but we're always going to be pumping it back out. And the same thing is true here for potassium. There will be an ion channel for potassium. That's always going to allow potassium to leak out. So there's always going to be some that the sodium potassium pump is going to actually carry back in. Now that's a transfer process. We also have something called endocytosis. Endocytosis are a collection of all processes that are going to bring materials into the cell. Some of the materials that need to be brought in would be, for example, fluid, large macromolecules, proteins, carbohydrates, and in the case of single-celled organisms like the amoeba, paramecium, large particles, which might be other microorganisms, as a way to eat. What happens is these materials are brought in, and they're trapped into little tiny vesicles, which are invaginations of the plasma membrane. They're going to hold those external materials. Now, endocytosis, I break it down into two types. Phagocytosis, or cell eating, because phago, first part up here, means to eat. So it literally says cell eating. This is the process of bringing in large macromolecules and large particles like other organisms. You know, so anything solid is going to be brought in this way. Now to bring in the fluids, we have pinocytosis, which literally means cell drinking. So now we're going to bring in the solutes and any kind of liquids. Now the next slide, I'm going to show you a short little video showing the process of phagocytosis.
In endocytosis, membranes invaginate, or pinch in, to form a vesicle, moving the enclosed material.
I want to stop it right here for a second to kind of demonstrate this. Notice how on this picture here, the material invaginated the plasma membrane or cell membrane. As it pushed in, part of the cell membrane forms around it, and this is your vesicle.
Inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
As explained in the video, this is a process common in many like white blood cells, like the macrophages. These macrophages are wandering around through your connective tissue, like in your dermis below your skin, and when they encounter a bacterium, they will invaginate and swallow up that bacterium, and using the lysosomes in the cells, actually break it down. So the infections are made harmless. Now the next slide, we're going to look at a short video of pinocytosis.
In pinocytosis, or cell drinking, the cell engulfs extracellular fluid, including molecules such as sugars and proteins. These materials enter the cell inside a vesicle. Epithelial cells in capillaries use pinocytosis to engulf the liquid portion of blood at the capillary surface. The resulting vesicles travel across the capillary cells and release their contents to surrounding tissues, while blood cells remain in the blood.
So now we just described two ways that solid and liquid materials are brought inside the cell. Well, the common feature among both of them is the invagination of the plasma membrane and the forming of a vesicle. Now, since materials can be brought into the cell, some cells actually expel materials, like on the next slide. In this slide, we're going to be looking at how cells expel materials. Exocytosis is the process where anything that's inside the cell is going to be pushed out. You know, so secretory cells, for example, cells that are going to produce hormones, this is how the hormones are going to leave the cell and get into the bloodstream. Now, we've already looked at one of these in our passive transport lecture, one of the contractile vacuole. might remember this picture of the paramecium. Well, remember, the paramecium is in a hypotonic environment. And what happens is this. The water flows into the paramecium and collects inside this special structure, the contractile vacuole. And when the contractile vacuole is full enough, it expels the water like a little squirt. Okay, but in the case of the hormones are produced inside the endocrine cells. And they migrate to the surface in vesicles, and once they reach the surface of the cell, they fuse with the plasma membrane and expel the hormones into the extracellular spaces. We'll show you this in the next slide, where you got a short video of how exocytosis proceeds. On this slide, you're looking at, the vesicle right here that contains, for example, a hormone or any other substance. Now, as you watch this vesicle is going to move towards the plasma membrane, where it's going to fuse, and these contents are going to be released into the extracellular fluid.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
Okay, so that's how we get materials out, in both the digestive system or the endocrine system.